got obsessed with art. I just love it so much how I can see the trees and the butterflies. And I got really excited when last year, my mom actually took me to see Asra, one of the most talented and amazing artists of our time in Connecticut, United States. However, when I got there, I actually got very sad and disappointed because it turns out that he wasn't really painting anymore. He was suffering from this grueling pain in his hands and his limbs that disabled him from actually doing the things that he loves and he enjoys. The disease he's suffering from is called sickle cell anemia. So because I did some more deep research into it because I really want to help him. I want to help him so much that I thought to myself, this disease must be caused by some very complicated processes in our body, and that must be the reason why we haven't found an effective cure for it yet. To my surprise, this is not actually the case. The disease has a very simple cause. It's just caused by a single gene mutation. So instead of having an A, you have a T. And because of this single change, instead of having a circular, disc-shaped red blood cell, you have a sickle cell. These sickle cells clutter together in your blood capillary and block your red blood vessel. And this is the reason why Asra and hundreds of thousands of people in the world today suffer from the pain and have to withdraw from the passion they have nurtured for so many years. What really bothers me about this is even though 300,000 to 400,000 people suffer from sickle cell anemia each year, we haven't found an effective treatment for them yet. Today, we're curing this using chemotherapy and blood transfusion, or bone marrow transplant. But the problem with this approach is only 10% of those hundreds and thousands of people can actually find a compatible bone donor. To a kid like me, I don't like this. This is not effective, and I want it to change. Because the disease is caused by a single gene mutation, I believe that what we should do is to take out the letter T, which is our wrong letter, and then insert back into our genetic code the letter A. Thanks to Jennifer and other very talented and brilliant professors and researchers at MIT, now we have CRISPR-Cas9. CRISPR-Cas9 is a new gene editing technology that essentially allows you to take out parts of the genes that are disease-inducing and inserts new parts of the gene you like. What is so crazy and mind-blowing about CRISPR that none of the previous gene editing technology can ever do? It is super fast and it is super effective. Can you imagine a disease that afflicted so many people can now be cured in a lab within only 2 weeks? This is not science fiction. This is reality. And we're actually in a very exciting time. 2 companies in Massachusetts, United States, CRISPR Therapeutics and Editas Medicine, are actually working on this approach. And the first clinical trial are going to be out this September 2018. So we're not far from that reality yet. I got really curious, and I want to get involved in CRISPR. So I want to learn how it works. We first need a plasmid, which is a circular piece of DNA. This plasmid contains 2 parts. The first one is the Cas9 gene, and the second one is the guide RNA gene. Once the plasmid is delivered into your cell, it is transcribed into the single guide RNA as well as the Cas9 mRNA. You can think of our cell as like a mysterious forest, and you want to find that genomic target. Our single guide RNA is like a very smart and brilliant detective who has all of the clues and knows where to find our genomic target. At the same time, Cas9 protein serves as this fancy Lamborghini that will take our expert there. Once this happens, Cas9 will induce a double-stranded cut to our piece of DNA and actually eradicate the gene that causes the disease. And our cell has its own mechanism of fixing it too. As a summary, you have the wrong gene in sickle cell anemia, and now you have single guide RNA and Cas9 serving as a molecular machine that will fix your gene. With this technology, we can cure a lot of genetic diseases we have today, such as Duchenne muscular dystrophy. So Anemia, one of the TCAS students who is going to present later, actually works with Dr. Ronan Cole at Sick Kids Hospital to cure Duchenne muscular dystrophy in mice. That is mind-blowing, because before, the mice weren't able to move, but only after 2 weeks, when CRISPR is applied, it can move without prior training. I got really curious into this, and I emailed more than 50 professors wanting to work in their lab. Not many of them responded, but then one of them, Dr. Westwood, at University of Toronto, Mississauga, responded to me and invited me to come to his lab. What we did with his graduate professors and also another students is we used CRISPR just to edit 2 genes in fruit flies, and we were able to double their lifespan. That is absolutely crazy. Can you imagine by using CRISPR and edit only 2 genes, you can actually double the lifespan of an organism? I just got really curious, and it was mind-boggling to think about how we can actually apply this technology to human, right? So I want to understand the underlying mechanism of this. I got deep into it, and then I realized that as our cell ages, its ability to resist against environmental stresses, such as temperature or toxic or chemicals, decreases. So logically thinking, if you want to increase the life expectancy of the cell, you need to increase its ability to resist stress. Now our question becomes, what is the magic sauce behind stress resistance in the first place? It's called heat shock proteins. I want to use an analogy to explain this concept. You have worked hard in your week, it gets very tiring, and you want to go party with your friends on a Friday night. And in that party, an accident happened because you got so drunk. So now, there are these supervisors and security guards who act as chaperones that help clean up your mess. Well, not the best thing that could happen, but thank God we have the chaperones. In our cell, a very similar process happens. Heat shock proteins act as chaperones for the proteins that get destroyed under environmental stresses. So the answer becomes, if we want our cells to live longer, we need to increase the amount of heat shock proteins. And we have 2 ways of doing so. The first one is to lessen the activity of the insulin IGF pathway. So this insulin IGF pathway we also have that pathway in humans. And what insulin IGF pathway does is growth promotion. So it takes up all of the glucose and all of the sugars you actually eat and absorb in your body, and it promotes the growth. It helps your body, the organism, to grow bigger and larger. But then the problem with this approach is our cell only has a certain amount of energy. If you dedicate too much energy to growth promotion, you actually have less energy for stress resistance. So logically, if we want our cells to have higher ability to resist stress, then we need to shut down this DAF2 IGF pathway and produce more heat shock proteins. So that's exactly what our lab did. We shut down the DAF2 hormone receptor, which is like this pump that takes in glucose and sugar molecules. The second way to do this is prevent the degradation of heat shock proteins. Within our cell, the protein also has its own life cycle, and sometimes it gets degraded. So my question becomes, how can we actually prevent this destruction of the proteins? Because we want proteins. We don't want them to get destroyed at all. It turns out that in the proteins, there's this thing called ubiquitin. So ubiquitin is a series of amino acids that act as a death tag for your protein. So once your protein is tagged, it will get destroyed. So now, because we don't want our proteins to get destroyed, we need to destroy the enemy of the protein, which is ubiquitin. To destroy ubiquitin, we need to mutate the gene that codes for ubiquitin. And by combining these 2 approaches together, our lab were able to double the lifespan of the fruit flies. Now, I want all of us to close our eyes and imagine a new universe of CRISPR-Cas9. Not only can we cure Duchenne muscular dystrophy, we can cure 6,000 genetic diseases that we're currently having today. We will not only see the lifespan of fruit flies double, but we can extend the human life expectancy by hundreds of years and allowing us to do things that we haven't possibly been able to do before, such as traveling to new stars, if that's something you fancy doing. with my expertise, talking to a lot of professors and working in a lab, I believe that that future will come. Thank you.